matters, phenotype matters, part of the talk. We talked about context. So what about the phenotype? If I could, get the fellows who are from my lab to pay attention. Anyway, so it's always impossible, right? So I hope that was on tape. So always, for us, the most relevant phenotype, particularly to get a grant, is the disease and the most closely disease-associated clinical measures. But the phenotypes that are most related to genotype and that are most understandable in a neuroscience sense are the ones that are closest to the DNA and to the molecules, the epigenetic patterning that may be determined by a promoter variant, the RNA expression, the protein sequence and function, small molecules that are the products of these proteins, and then this intermediate level of brain circuitry and cognitive measures, where still the allele effects are stronger than they are at that level of the complex disease. So what's the evidence that that nice little model is actually true? Well, here was one of the first imaging genetic papers. This is from the Danny Weinberger lab. I was involved in this study. It involves the serotonin transporter promoter polymorphism. It has two major alleles, the smaller allele, the S allele, and the L allele. And the shorter allele is associated with lower expression of the serotonin transporter. And it had been observed for a number of years to have modest effects on anxiety and emotionality. But it has very profound effects on the response of the brain to an emotional stimulus. In this case, the emotional probe is the faces paradigm, where you're shown angry and happy and fearful faces. And to evaluate those faces, one activates the amygdala, and the level of activation is actually predictive of subjective emotional response. It does correlate with what happens in some anxiety and dysphoric disorders. And you can see that these significant effects were observed in very small matched cohorts of individuals. And so you have 4, 5, and 5 individuals with the different genotypes and matched for age and diagnosis and other parameters in one cohort here and another one in another cohort here, and with the low expression allele of the serotonin transporter predicting stronger fMRI activations after the emotional probe. So that was one of the first evidences that this imaging genetic paradigm looking at intermediate phenotype worked better. And the serotonin transporter neuroimaging literature has developed way further. And in papers that I was not involved with personally, here is one of the 2 papers that appeared in Nature Neuroscience 8 years ago, showing that what the transporter polymorphism is probably doing more profoundly is altering the connectivity of frontal cortex to other regions of the brain, including the amygdala. So it appears to be a connectivity gene, and it also alters structure, as shown in the original paper with Ahmad Hariri. The amygdalas of people with those SS genotypes were smaller than in people who have the other genotypes. And these imaging findings have been replicated in most hands and seem to provide a way of understanding how this polymorphism actually works to modulate the complex trait of anxiety. And that took a number of years, for the different levels of analysis to be worked through for the serotonin transporter. Increasingly, people are trying to do it all at once. And so instead of producing one part of the story, such as an association of the variant to the behavior, and then somebody else associates it to an imaging phenotype and somebody else to the molecular change, why not do a multi-level analysis? Well, of course, it's not easy because you have to pull together a number of different collaborators and make a major investment in the paper and in the study. But it does become, and also you have the splitting of credit. But I think it's very important because then we don't end up with so many of these findings that are, on the one hand, very often never replicated, but on the other hand, even worse, never explored at these different levels. And so there are hundreds of association findings in the genetics behavior literature and the genetics addiction literature that have never been explored at other levels that they really should be, and that's because it wasn't necessary to publish the paper in the first place, and then maybe the group split up, somebody got another job, whatever happened, and then it just never happens. And so here we try to do a multi-level analysis of a functional locus that we found at the neuropeptide Y gene. Neuropeptide Y is an anxiolytic neuropeptide. Many of you may know that its levels predict PTSD and war veterans and others exposed to stress, and so it's an important neuropeptide we already know in the stress axis. And so taking this functional promoter variant, we were able to look at different phenotypes, starting with the effect on RNA expression, where the variance accounted for is high. It's about 28% of the expression of this NPY RNA in lymphoblastoid cell lines from people with the different genotypes. And also there's a strong effect on the level of the peptide in plasma from controls or patients, 17% of the variance, 6% of the variance. And by the way, Regita was also involved in this work. She is the one who actually contributed the healthy controls and alcohol-dependent patients on whom she had actually measured NPY, and so the availability of this molecular phenotype was crucial to really seeing this whole pattern of effect of the locus all the way from molecule to behavior. So then here in the middle are the imaging genetic findings. There were two different imaging genetic studies where, again, there's a large percentage of the variance accounted for. These are looking at emotion responses of the brain for stressful emotion, and also a pain stress paradigm where the low expression NPY genotype is predicting a high response of the brain. And now the subjective measures, affective response to pain, pain threshold, trait anxiety, the effects are small because these are finally the emergent behaviors which are altered by so many things, including NPY, but of course the whole history of the person and the way that they choose to subjectively interpret the events that are going on in their own brains. It's very much like if we all had with us a little portable, it's just an iPhone, a little portable scanner where we could read what was going on in people's brains. And we were all outside in the bright sunlight and happy just a moment ago, and if we'd scanned everybody's brains, we would have seen happiness. And now we're back here and listening to David Goldman, and so it's definite trough. And so ask people, how are you doing? They'll say, I'm doing fine. And you have your little scanner, and you say, well, I'm sorry, but that's not what the scanner says. But there is this problem that even though the thing is happening in the brain, we don't always know that it is going on. And that's why we have our scanners. Here's one of the imaging genetic findings from that NPY paper. And you see that it's, again, a relatively small neuroimaging data set with the 3 functional genotypes. This is the lowest expression genotype, and the fMRI is activating maximally in amygdala in hippocampus when Ahmad Hariri gave the subjects the emotional stimulus. And then the higher expression NP-wide diplotype, they're activating less, or almost not at all. So there are many findings of that nature now that have been achieved with imaging genetics. And so one might then ask, well, why not then take some of these genome-wide association studies and explore the neuroscience basis of those with imaging genetics? And also, why not do imaging genetics in the first place and then do genome-wide association? And this is exactly what's happening all over the world now. There are two very large consortia, one Imagen and the other Enigma, in Europe and in the United States, that are doing imaging genetics, GWAS. My lab has published some large intermediate phenotype GWAS, which I'll not be showing you today, but on EEG, where we found multiple genome-wide significant loci. And so these can be better and alternative phenotypes for GWAS, much smaller samples, and I think finding new genes, but then also used for in vivo functional genomics follow-up for GWAS hits and to understand mechanism. And so I'm going to go back to the CHRNA5 variant. This is, as Marina said, the Mr. Big GWAS hit from the nicotine GWAS. It's been very extensively replicated. This 398 ASP allele has a role in smoking vulnerability and thereby probably, but maybe there's another mechanism, a role in lung cancer. But the attributable risk is less than 0.5% of variance. So it accounts for very little of that heritability of nicotine addiction that I showed right at the beginning of my talk. There must be many other genes involved in addition to this one, but it is a functional locus, as Marina said, and we were interested, Elliot Stein and Elliot Hong and us, in whether that unfortunately, some of the p-values here have been obliterated, but they're significant. These little number symbols have substituted for them here because of the lack of space. We were interested in whether that that it could affect a circuit that Elliot Stein had identified that appears to be important in nicotine craving. This is the Fagerstrom score, which is a nicotine craving score here on the y-axis here on the right. And he had measured functional connectivity between different regions of the brain. And this is he used a different method than Marina described. Instead of using diffuser tensor imaging, Elliot Stein here is using resting state functional connectivity, whereby one just looks at the moment-to-moment oscillations in electrical activity of different brain regions and looks for correlations in activity, and thereby infers that there is a functionally connected circuit involved there. So you don't really know the pathway of connection of these different regions, but you know that they're related in their functional control in one way or the other. And so here he has correlated this functional connectivity with the lower functional connectivity associated between dorsal anterior cingulate and ventral striatum associated with higher Fagerstrom score. And that finding is a new data set where that's just been replicated, and we've also replicated the association to the ASN398 allele that I'm going to show you here. So here then is the genetic data on the left side. First of all, even in this small data set, this allele is associated with smoking risk. So if you have the allele, you're more likely to be a smoker, even in this quite small data set of only a few hundred people who were brain imaged. But the real point is to look at the effect on functional connectivity, and it is this risk allele ASN398 that predicts lower functional connectivity, and this effect is much stronger than the effect of the allele on smoking risk in the general population. It turns out that it doesn't just affect functional connectivity of dorsal anterior cingulate to striatum, though. It affects or predicts functional connectivity just as well for 7 different brain regions. 7 different brain regions whose RSFC Elliott Stein and colleagues studied. And so it well may be that even though it predicts the connectivity of Elliott Stein's favorite circuit, that what's really important is its effect on that habenular circuit of Paul Kenny's, which Marina was describing earlier. We don't know that because the resolution of the imaging is not sufficient to resolve that circuit at this point, but I'm going to be bothering people until they construct some sort of billion-dollar machine that will enable that to be captured. Yes?
So I have a question about how that connectivity data
Yes.
is really, like, how it's determined. So so they're measuring the activity in the brain, and then they just kind of randomly try and correlate every possible pixel in the image?
No. Now that's, in fact, that has been done. There are now, shall we say, unguided functional connectivity maps, and from those maps, there are several meta circuits that have been generated, and some beautiful papers which show graphically these meta circuits. So they include, for example, the so-called default network, and there's several other networks that have been identified that way. But in this study, what was done was to seed the analysis by pointing to a particular voxel and asking what other voxels in the brain predicted that voxel. So it can be done in the unstructured way that you're describing. Is the way I like, but generally it's done more in this guided way. And there's a lot of the imaging analysis that is not done in a totally unstructured way, by the way. When you hear about fMRI studies, a lot of those studies where you see that there's just one region that pops up with a really nice signal, and all the rest of the brain is like black. Well, sometimes that's because they masked the rest of the brain in their analysis. So you have to be very conscious of how they've done their statistics, because to some extent it's a similar problem as with the genomics, which is that you can imagine that there are a lot of different permutations and possibilities possible when you think about all of the two-way voxel to voxel combinations that are present in the brain. And so I think that this is going to be an incredible area of progress. On the one hand, you want to see it done so that you would find everything. You would find what you don't expect to find. And on the other hand, you certainly don't want to throw away or discount what we already know about brain circuitry and connections. So it's a little bit about, and I could say the same thing about the genomics, which is that there are people who want to take all of those genetic markers and just put them into a statistical program as if each marker had no relationship to the other markers. And I'm opposed to that. I think that you have to take into account the linkage disequilibrium relationships of one marker to the next, and not all just independent entities. You want to know about their physical relationship, the possibility that some of them are co-regulating the same gene, altering the same gene product, and then there's this whole other direction of then taking the multiple findings and trying to use them to understand if you're observing common perturbations of the same functional network, like are all these genes related to malinaldehyde metabolism or to synaptic function. And those are very important approaches, and it's a continuing tension in how to do omics studies, whether to do them unguided, semi-guided, or completely guided. And we don't really, and I think it kind of all needs to be done. It's a great question. Okay, so here are conclusions for that SNP. It's a validated variant, but it accounts for very little of variance in addiction. There's a risk allele that more strongly predicts weakness of some circuits in the brain. the weakness of one of these circuits predicts craving in the Elliott-Stein hands, although the Paul-Kinney circuit is apparently a different circuit. And this speculation that I had here that what this thing may be doing is not altering affinity for nicotine, but altering function of circuits involved in craving. So now I'm actually to the second part of this talk, and I can take it a bit faster. I'm looking at the schedule though first, and I'm going to try to wrap things up at 2:30, Chris. Okay? So I'm going to talk about finding rare and uncommon variants by deep sequencing in people and in model organisms. And there are different types of application of this next-gen technology. We've already heard, by the way, about several of the other applications, especially the ChIP-seq application, so I'm not going to talk about epigenetics today. But mention that there are these different types of resequencing. One can go after single genes, for example, BRCA1, which is what Myriad Genetics is in the business of doing at $800 an analysis, or was until the Supreme Court decision. Or one can do small gene panels. One can sequence chromosome regions, and you might have to do that if you feel that you have a linkage result that the locus must be in that chromosome region, and you've already ruled out that it's within the coding sequence of the genes in that region. And so just sequence everything. Then the large gene panels, these are very easy to do now with the adjuvant capture arrays and other technologies where you can pull down sequences of 500 genes or all the genes on a chromosome if you want. Whole exome sequencing is right now the workhorse sequencing technology in clinical genetics. There are actually thousands of patients who have mental disability and other syndromes who are receiving whole genome sequencing year by year now. And hundreds of them in whom probable causal variants are being discovered. These are rare, predictably severe variants that are being found by whole exome sequencing. So the exome is this so-called expressed region of the genome. It's usually a 50 megabase target. Your genome is 3,000 megabases. Everybody who read the newspaper knows that ENCODE proved that 90% of the genome is functional, and that most of the important function is in regions of the genome that are not in genes, et cetera, et cetera. Wrong. What happened was that ENCODE defined as functional anything that was expressed at whatever level. So if there was one RNA expressed in one cell, then by the ENCODE definition of function, it's defined as functional. And so the problem is that there are all kinds of ways that transcripts are made, kind of by mistake, or this or that, or maybe it's important, in a cell here or there. But today, even though 90% of the genome could be functional, if it's in that 90%, we have very little knowledge or ability to predict what it does. And so instead of sequencing the whole genome and then throwing out that 90%, what most of the clinical genetics community is sequencing this 50 megabase target for the reason that at least some of the variation in that region, not all of it, but at least some of that variation, we know how to evaluate whether it's functional or not. So it's not an issue that there's not functional variation elsewhere. It's that the ENCODE results were way over-exaggerated, and that as a practical matter, we don't really know what to do with the data for the most part today. And so instead, do some molecular computing and just sequence the exome, and that still costs $800 a sample, and whole genome is 5 or 10 times that much. And so that's the explanation for why this whole exome sequencing exists today. And if anybody asks you, are you better off having whole genome sequence or whole exome sequence? Of course you would want the whole genome sequence, because more is always better. But don't expect that your bioinformaticists will be able to do anything especially intelligent with most of it. I mean, there's a, that, like, let's say that even that you're looking for deletions. If the deletion doesn't involve a genic element, you're still in trouble. And finally, what about genotyping? Well, genotyping is actually a form of sequencing. And this is especially obvious if you're in a lab that does lots of analyses of this sort. So, sometimes if we find a new variant and we just want to genotype it in 100 samples, we'll just do Sanger sequencing to genotype it. But also, when you are doing most forms of genotyping, you're interrogating a small number of nucleotides in the genome, and if you do 1 million of those genotypes, you're interrogating for sequence a large number of genotypes, a large number of nucleotides. Obviously, if you genotype 1 million markers, you're interrogating at least 1 million nucleotides of sequence.
David,
Yes?
what is the rate of somatic mutations?
Good question. The rate of somatic mutations is high. And the beauty of it is that when you have a genomic DNA sample, you have a mixture of genomic DNAs from thousands of different cells, which all have different somatic mutations. And so those all just work out to be the background noise, just like sequence error in your sequencing result. But in particular projects, you're focused on somatic mutations. For example, we're doing sequencing right now in esophageal cancer, and others who are doing cancer sequencing are concerned or totally targeted on what are the specific mutations in that tumor. And those tumors are clonal. And because they're clonal, then you see the somatic mutations. And I can tell you that in the esophageal cancers, that in that 50 megabase target, that we're seeing 4,000, 5,000 somatic mutations, most of which are not connected in any way to the actual cancer. So there's a lot of somatic mutations in a clonal line. And there are all kinds of things happening in individual clones of cells. When you go from neuron to neuron in the brain, if there are 2 alleles at each autosomal locus, from neuron to neuron, there's a good chance that one neuron is expressing one of those alleles and the other neuron isn't, and it's expressing the other allele. There is an enormous amount of somatic mosaicism, both at the genic level due to somatic mutations, and also at the epigenetic level due to the arbitrary shutdown of one of those 2 copies of the gene or the other. And that's been proven by actually looking at clonal cell lines from the same individual. And you can see that there's one allele or the other that are flipped in their expression. There's a paper in the latest Nature or Science which has a method for visualizing the somatic mosaicism of expression at the single cell level, including in the brain. It's just out.
So if you took monozygotic twins,
Yeah.
did their blood, and how many differences do you see?
Well, MC to MC twin, if you're averaging across, averaging...
There's stem cells which are going to mutate as well.
Actually, I don't know the answer to that question because there should be mutation at that level, at the stem cell level, and then carried through all the way. I think it has been done. I think it has been done, and I actually don't know factually what the result is. My prediction is that they found thousands of differences, genome-wide. And of course, most of those differences landed nowhere. They landed in, these spaces that are, called by ENCODE to be functional according to their definition of expression, but they're probably not doing much. Okay, moving on. All right, so now we're going to move on to impulsivity. And there's this simplistic definition, action without foresight, but we know now that it's much, much more complicated. I mean, there's the motoric impulsivity, and there's the evaluation of risk type of impulsivity, and there are many experimental measures of this in humans. and in animals. And what's fascinating is that a lot of the different measures really do let you make some correspondence between the phenotype or the type of impulsivity in the human and that in the animal. But I can't go into that in any detail. Impulsivity is a key mediating trait not only in the addictions, but also in other psychiatric disorders. For example, consider antisocial personality disorder and ADHD. The way that the genes have mapped onto these impulsivity constructs is not the way that they've been constructed by behaviorists and neuroscientists, however. The genes don't really behave themselves in that way. They are acting pleiotropically. They do many different things because most of them are expressed all over the brain. And so if they're altering motoric impulsivity, there's a good chance that they're also altering risk evaluation and other behaviors. And so, you know, we're not, as we find the genes, going to see this, you know, this perfect type of mapping, you know, like we found the gene for the id and the ego and the superego, which is not going to work that way. And so Brunner described a syndrome, which is called Brunner syndrome, in one Dutch family, and it's due to a point mutation. This stop codon in monoamine oxidase A. MAOA and MAOB are tail to tail located on the X chromosome, and as you know, they metabolize a variety of monoamines, and this one Dutch family had come to Brunner and colleagues really demanding to be studied. They had this behavioral syndrome, call it what you will, with discontrolled behaviors, including aggressive outbursts, arsons, attempted rapes, and exhibitionism, not all in the same individuals within those families, but within the family, but you know, one of the other behaviors. And here the affected males are blacked in here, and you'll notice that there are no affected females. No affected females. And so this was a pattern of transmission that would be consistent either with the sex-modified trait or X-linked recessive transmission with carrier females who are unaffected and affected male offspring. About half of the male offspring of an infected female would, of course, be affected because, you know, they'd have half a chance to get the bad X chromosome. And the study was conducted during the time when people measured monoamine oxidase A and they measured monoamine metabolites. They measured them everywhere that they could. So standard NIH study was that you get fluids from all sources in the body, the anterior chamber of the eye, if you could. And then you take these fluids and they'd be measured for the monoamines and their metabolites. And so when these metabolites were measured, in these cases, there was evidence of abnormal monoamine metabolism. Put that together with the fact that there's an X-linked pattern of transmission and the gene is on the X chromosome, and Brunner, Xander Brakefield, and colleagues figured out that they should sequence the MAOA gene. When they did, they found the stop codon, and also they found that there was no fibroblast MAOA activity. And so that's an example of a complex behavior actually parsed right down to the level of the functional locus. It led to that Caspian-Moffat G by E. It's led to imaging genetics papers, for example, from Andreas Meyer-Lindenberg. You know, so we know that there are more profound effects of the MAOA on brain function. And the reason why it could lead to that was because in addition to the rare variant, there was the common variant found at the same gene, this common short tandem repeat polymorphism. It doesn't have as strong effect on function as does the stop codon, of course, but it does lead to low expression of MAOA. And here's our gene by endocrine interaction study with MAOA. It's known that males are more aggressive and impulsive in general, and it's known that within males that higher testosterone levels predict higher aggression and impulsivity, which is, you know, not real comforting when you turn on the TV every night and there are advertisements for 50-year-old males to receive testosterone supplements. Let's give them testosterone. Meanwhile, females are actually down here at below 50 picograms per mil of testosterone in their CSF. So this is the relationship that had actually been seen for a number of years, but it's only in individuals who have the low activity MAOA genotype, people who have the high activity MAOA, and that's a lot of people, a lot of males, did not show an increase in aggressive and impulsive behavior. So we've been looking for other strongly functional rare variants and trying to do this in powerful contexts. What was special about the context that Brunner used? What was special was it was a single Dutch family. That stop codon was never found again. And so if you had studied it in the population context, like collecting 10,000 individuals in a GWAS, you would have still just found one person at most with the stop codon. And now you would be forced to say, is this person more impulsive because of this stop codon or because of something else? And that would be kind of hopeless. But because Brunner had this pedigree, it was in this advantageous situation that the frequency of this stop codon in this pedigree, because they're males and females, is about 33%. So instead of the stop codon having a frequency of 1 in a million, it has a frequency of 1 in 3 in this family. I mean, it's an allele frequency, and you can see that in this family that you know, just about everybody is at least a carrier of the stop codon, except some of the males. So the other thing that one can do is to search for genetic effects in other contexts that are powerful. And so the two contexts I'm going to tell you about at the end of this now are the Finnish founder population and then some selectively bred rats. And so here are the Finns. This is as visualized by genetic ancestry analysis using hundreds of thousands of genetic markers and by the NIAAA group. And you can see that, that with a eigenvector type analysis, that there's a gradient of relationship of Europeans from Southeast Europe to Northwest Europe. Populations in Northwest Europe are more, those individuals within those populations are more closely related to each other than they are to individuals in Southeast Europe. And here are the Finns who are sticking out as an individual population. And so that's one way that you can look at a population such as Finland as being a distinct population, but more important is the fact that this is a distinct population that has evidence of founder effects for medical genetic diseases. The Finns have, because of a population bottleneck at the founding of Finland, and also another bottleneck that occurred when Finns were sent off for the 100 Years' War, the Finnish males, and many of them never returned, and also there were terrible battles fought in Finland. There was population bottlenecking, and it dramatically reduced the genetic heterogeneity for a number of medical diseases. There are multiple diseases where there are 8, 10 genetic forms elsewhere in Europe, and where there's one form of the disease in Finland. So you can hope that there would be, kind of like in a large family, a powerfully acting genetic variant that could have come to a high frequency in Finland due to the founder effect. And so we did find such a variant. This was found just by sequencing 14 different dopamine and serotonin receptor genes. And this was the stop codon that we found in the HTR2B serotonin receptor. And you can see that it truncates the protein product just 20 amino acids in, and so you lose the elegant 7-transmembrane domain GPCR structure. And the variant is effective as a stop codon. It leads to, in heterozygotes, a 50% reduction in the amount of receptor protein. This is a Western blot. It also leads to something called variable nonsense-mediated decay, where if a RNA has a stop codon, that it's subject to an enhanced decay process in the cell. And so you can look in the paper and see, if you want, that there are many people who carry this variant who have low levels of the HTR2B RNA, and that's because of this nonsense-mediated decay, we believe. And then there was association of this stop codon in the Finnish population, comparing cases and controls, and also co-segregation in a limited number of these Finnish families that we had, where we were able to follow antisocial personality disorder and alcohol use disorders, and there's significant co-segregation of the stop codon with the behavior in a similar fashion as Brunner did with his one family. So what are the behaviors of these carriers like? Well, we had originally screened 96 severe impulsive criminal offenders, compared them to, and then sequenced also 96 Finnish controls. So those criminal offenders who we found this thing in and who we subsequently found it in were cognitively normal, and their crimes were impulsive, and not for any recognizable gain. 3 double murderers that were in the sample all were carriers of the stop codon. They were also more likely to attempt suicide. They had normal CSF monoamine levels. It's not an ancestry informative marker. Overall, the frequency of the stop codon in Finns is about 1.3% to 1.4, maybe 1.5%. So there are over 100,000 Finns who carry it, most of them completely normal, which means that it's a factor in impulsive behavior, because that's what the family co-transmission data show, and that's what the case control association data show. But just observing the fact that most Finns who carry the stop codon are normal, it's not a sufficient factor to explain violent murder or other discontrolled behavior. It's a contributor, and it may be that the effects are stronger in a particular family context, such as when we were following the potential co-transmission of the stop codon with impulsive behavior in a family. This is the knockout mouse for HTR2B. It has stronger novelty-induced locomotion, whether in the light or in the dark. It has stronger D1 agonist-induced hyperlocomotion. It interacts more often with a novel object that you place in its cage. So you might say it's a novelty-seeking gene. This is the delay discounting task that we've been discussing, and you can see the divergence in delay discounting of the HTR2B knockout mouse, where it's not willing to wait as long for the larger reward. And then finally, the hyponeophagia test, the mouse is made hungry by food deprivation for 24 hours. The food pellet is put in the middle of the cage where it doesn't want to go. Ordinarily, the mice wait about 3 minutes before they go to the food pellet, and these ones wait about a minute. And so this is the example of what happens when an American geneticist kind of has this, kind of crude, direct way of thinking, works with some elegant French neuroscientists, Luc Maritot and Stéphane Dolly, and what happens, Brigitte, is they promptly generate an image like this, which is somehow they made this. I don't know how they got the 2 mice to behave in this way in the clock. Actually, I think it's cut and paste, but here is the mouse with the 2B receptor, and it's waiting patiently for its to satisfy its hunger, but the 2B knockout is right away gnawing away the bezel of the clock. So Luke has done, together with us a little bit, Luke has done a lot of neuroscience extensions of this HTR2B finding, and then we've done a lot of further work on the genetic side. So we found that it is highly abundant in fins. So we've now genotyped with colleagues in Finland over 20,000 fins, and it has this high allele frequency there, but it's not found really anywhere else in the world. Luke has shown that it's co-localized, that it's localized in 40% of VTA dopamine neurons. It's regulating dopamine release. It's modulating cocaine-induced hyperlocomotion. We've worked quite a bit now on a haplotype, which is predicting cocaine addiction and also is predicting dopamine release in a neuroimaging paradigm involving stress. That's done with Shankar Zubieta. But I'm not going to go into all of those details. I'm just going to go to one important conclusion on this slide, which is that it's been known that deep sequencing can work for Mendelian disorders. If you have a Mendelian disorder, such as a monogenic kidney disease or whatever it might be, and you collect a number of people who are affected and unaffected in a family and deep sequence them, you can identify the causal variant because it's a Mendelian disorder. But we believe that we're now in an era where the deep sequencing can detect functional alleles that are also linkable to common and complex diseases as well, but it depends upon contexts. And we don't have all the context that we would want, so we've gone to some of the animal models that seem to also alter powerful context. And the animal model that we first went to is the alcohol-preferring, non-preferring rat.
So before you
Yes.
looked at different genetic backgrounds and modifiers of the knockout?
No.
Systematically?
No. But I can tell you this, that except for one thing, that Luke Marito has his HTR2B knockout, and we also have an HTR2B knockout on a different background. And there's a dramatic difference in another problem that is associated with the stop codon, which is congenital heart disease. Our homozygotes don't survive. His do. And so that is a genetic background effect, and it's probably specific to the congenital heart disease problem that accompanies the HTR2B knockout. Yes?
So this is kind of more of a big picture question. Now we have this information,
Mm-hmm.
that these individuals are susceptible to the disorder because of this mutation, and we could maybe start to think about therapies. However, a very small fraction of the people who have the mutation are actually manifesting the disorder. So what do you do? How do you think about treating that? Do you say you take medicine preventatively so that you don't have impulse break out? What do we do with this type of information? Because it seems so powerful.
I think that the most powerful predictor of behavior is not some combination of genetic markers, but it's observation of behavior. So the better way to use the information would be in people who have self-identified themselves as having a problem, or who, when evaluated on some test which is strongly relevant clinically, like it might be a measure of frontal function, executive cognition, maybe they have very poor executive cognitive function, and they're having problems in multiple areas, and they don't recognize it themselves. But where they're already there, then you begin to ask why, and part of the why would be their genotype. Good question. It's certainly relevant with regard to a variant such as that aldehyde dehydrogenase 2 variant, because we know that if you have it and you flush when you drink alcohol and you going to drink moderate amounts of alcohol, that you're going to put yourself at a higher risk of esophageal cancer. So shouldn't there at least be some sort of counseling provided to people? There is no such counseling today. There's no information out there. I mean, there are a couple of articles that we've written, but my institute is not taking this up as a jihad. They're meanwhile advising people that moderate drinking may be helpful.
Because it kind of leads to a more philosophical question of, okay, are we going to genotype everybody, and then if you have these mutations, then you need to seek counseling because you're at high risk, or you wait until there's been a problem, so we know how to treat you because we have this correlation in our database.
Yes, I'm with you on that. I predict that within a decade that most people will have had their whole genome sequenced because costs are coming down and there's so much information there. I'm hoping that's what's going to happen. I'm hoping it's going to happen because I think that it'll be such a trove of data that we'll find a lot of relationships between genes and phenotypes that we don't appreciate now. So the model that at least one company has, their business model is that every child will be sequenced at birth, and of course that they will do it for a fee. That seems like an efficient way of doing it to me, because then it's done once and for all, and you have that information. If it's in the right database, then whatever the gene might be, it might be BRCA1 that you might be concerned with, or CFTR, the cystic fibrosis gene, or whatever, that it could be accessed, and one can do just what the brain imagers do. One can choose to mask portions of one's genome. So if you don't want certain information, then that can be masked, and that's a huge controversy today. There is just out in Science a couple of weeks ago a point-counterpoint based upon the American College of Medical Geneticists' recommendation that 57 inadvertent genome sequencing findings be reported, regardless of whether the people who had the sequencing performed had requested that information. So what would happen is that you might take part in a study, or you might have had sequencing done for one particular problem, but along the way your whole genome sequence is determined, and it would be found that you had one of these other 57 problems. And so what the American College of Medical Geneticists has recommended is that information be transmitted to the person, even though the person never asked for it. So, I hope I didn't give away my opinion by the tone of my voice, but let me just say that the conflict here is between these two crucial imperatives that we face in all clinical medicine and in our human research, and those are the principles of beneficence, where we want to do good for people, we want to do what's best, but also the principle that we treat everybody as the principle of autonomy, that we treat each person as an autonomous moral agent who makes their own decisions about things like that. So, in an era where we're moving towards having whole genome sequences available, these questions are already upon us, even if we don't want them to be.
That's already a big question if it's your own genome, but what if it's your family members or your twins, your identical twins' genome? So the family of Henrietta Lacks, who was the woman who HeLa cells came from because of her aggressive cancer, sued to stop the publication of the full genome sequence of the HeLa cell.
Yes.
Because it invaded their privacy, and she had not consented, of course, but neither had they consented to have their family genomic information publicly available. they've come to an agreement that that will be in a database and that the purposes for its use will be cleared through a foundation that family members will sit on. But it doesn't have to be the case that it is such a famous cell line. It could be that you have your genome sequenced and your kids really don't want to know.
Absolutely.
That could be in the public domain and
Yes.
that's it.
Are there treatments for all 57 of those conditions that they want reported?
No.
So then what's the point of telling the patient if they're...
That's the Huntington's.
Yeah, like what if they don't want to know?
Exactly.
Many cases of that.
And the
In
Alzheimer's
each instance, there's a separate argument as to why the information might or might not be valuable, and why under the beneficence imperative that you might want to give the information to people, and in each instance there's a reason why a person might not want the information.
There's also...
Yes, Santo.
Sorry, can I make a comment, Jessica, on what you just said? There might be some reasons.
Right, even though there's not a treatment, you might want to know for Huntington, you may not want to have children. Yeah.
So there might be some
You wanna go on
other
big
reasons, because, for example, you have certain clinical trials that are ongoing at NIH. NIH, the hospital, is a perfect example. And you're eligible to fit and to enroll into those clinical trials only if your disease has a certain degree of severity. For example, they're looking for specific patients. So knowing, for some families, I do have a family in Italy who has Huntington's disease, they really wanted to know how many repeats were screwed up, basically, because they wanted to make plans for their children, grandchildren. So maybe if you know you have a very mild form of certain disease, you don't worry, but if you are, then you start becoming extremely proactive for your family in looking into what are the possibilities in terms of experimental therapeutics and so on. So some people are really,
Some people want, some people don't.
absolutely,
So there should be that choice.
exactly.
One more on this.
Yeah, there are examples like Huntington's and such where genetic variants, the predictability of those are quite good. But my worry is that there's so much, if you take like all GWAS associated SNPs and try to make predictions off of those, that it's, you're providing a lot of misinformation out there. And I could see that as unethical, like if you're trying to predict, well, I think addictions fall under here, you know, if you look just at your, you know, SNPs that have been associated, you know, that prediction's just so poor, and, you know, people may be changing their behaviors for the good or bad based on those, and there are a lot of problems, I think, as it stands today. A decade from now will probably be a very different boat, but. I think even if the information's accurate, that it just leaves you wide open for things like discrimination and stuff like that based on your genome. Like, what if we get genist about people? You know what I mean? That's like Gattaca, the movie.
I do. I have the. I often show the Gattaca slide with Ethan Hawke and Uma Thurman. Everybody should see Gattaca.
Yeah, I agree.
Okay, so we applied it to the P and NP rat. It's a pretty good model for alcoholism. It was selected for 70 generations for the high and low alcohol consumption. They drink till they're drunk. They sustain high blood alcohol concentrations, consume it for the CNS effects. They show alcohol withdrawal when it's removed, and they show the important relapse-like behavior where if you deprive them for a couple of weeks, they will rapidly reinstate to high levels. And we exome sequenced 6 of the NP, 6 of the preferring, and 4 of the parental Wistar, for the parental strain rats, which are Wistars. And so what is the yield of a sequencing effort like that? Well, it's a 50 megabase target. There are about 120,000 SNVs, or single nucleotide variants. There are 20, and so then you have a bioinformatics pipeline to parse those, to deal with this massive amount of information, because you can't study 120,000 things. There are 25,000 of them that were completely segregating or homozygously different between NP and P. And so we arbitrarily decided that we would only focus on the segregating SNPs. There were 800 of those that were missense variants. That is, they altered coding sequence of proteins. So we might hope to understand their function today with the bioinformatics predictive programs that we have. We ran those predictive algorithms, and 38 of the 800 are predicted to be damaging, including 2 segregating stop codons, so we stopped there. We stopped with the 2 segregating stop codons. And obviously, and I will tell you, we are working on other variants. There are many of the other variants that are quite intriguing and that we think alter alcohol preference, but those stories will be slower to develop. Here are the 2 stop codons. One is in the metabotropic glutamate receptor 2, and the gene is located here on chromosome 8 of the mouse, and the rat. And here's lipocalin 2 on chromosome 3. They're both in large fixed blocks. So after analyzing many animals, we found that there are these large blocks, which are apparently the consequence of early selection, because there was a rapid response to selection in these strains, and then there's a problem with random fixation, which I'm going to show you in the next slide, but then there was early fixation, actually, of these blocks between the P and NP rats, and we think that this is a way of finding some additional loci. So they are in some fairly large fixed blocks. They may look small on this slide, but genomically, those are millions of base pairs. So here's the problem of random fixation and why, for a long time, people never got anywhere with their selected strains. They would select the high and low strain, and then they would find that their high strain was different in some parameters compared to the low strain. But perhaps that was by chance, because there are many things that are being randomly fixed when you have a small breeding population. And you can't maintain thousands of animals. They maintain 40 pairs, usually, in a rotating mating scheme in a large selection experiment. And so the consequence is this. This is across the exome. This is the amount of genetic variation is measured by nucleotide heterozygosity in the Wistar rat. They have a generous amount of nucleotide heterozygosity, about 1 variant per every, whatever it is, base pairs. That level of genetic variation is reduced by about half in the P rat and to about 1/3 in the NP rat. And so if you were just focused on any random candidate gene of interest, there would be a good chance that it would be randomly fixed between P and NP. You have 2 points. 2 points always make a line, and so you say we've discovered the cause of different alcohol preference between the P and NP rat. And same thing if you had measured some aspect of physiology that the P and NP rat happened to differ with also. You just made an association based upon fixation. But we found that almost all of this random fixation occurs not in the large blocks, but just randomly all across the genome. So most of the random fixation is, first of all, dispersed, just here, there, and everywhere. And second, we were able to genotype F2 animals in which the genetic background, all of that random fixation, has been scrambled up again so that you can look at the effects of an individual allele against all the different genetic backgrounds in removing the problem of random fixation. And so here's how that's done. The inbred P and the inbred NP, which still differ in the behavior, are crossed, making F1s. And then F2s are made by crossing the F1s. Meiotic recombination occurs here, scrambling their genomes up into mosaics of P and NP DNA. And then you measure the phenotype in these, in this case, 384 F2s that have been genotyped. And that led us to be able to show that the animals that were homozygous for the stop codon, there were a lot of them, there were like 90 or 100 of them, had a 28% increase in their alcohol preference, independent of all this other genetic background. And there might be gene-gene interactions too. We don't know that. Yes?
So, just one
Yes.
point that I think you want to make in terms of if you're going to do selective breeding at the get-go, you might want to have 3 independent lines.
Absolutely. And that has a lot of virtue, except I've been worried about it lately. And you might get more worried about it, actually, if you look at the new rat genome sequencing papers, which have just appeared in Nature Genetics. The Rat Sequencing Consortium has published a wealth of of rat genetic variation, which is that there's some of the variants that are relatively common, and if they alter the trait, they would then be subject to the selection. And there are other ones that might be a concern because if they're rare enough, just by happenstance, the first few pairs that you started your selection with might not have it. So it might not always work. But I agree with you that replicate lines should be done if you're starting.
But you can still do the F2s, if, you know, if.
Yes, and you can still do the F2s. And so here's the effect of the GRM2 stop codon on function. You know, we were homed in on this like a laser beam because it's a metabotropic glutamate receptor, and it regulates glutamate release. And so this is the Western blot. P rats with the stop codon lack the protein. And here's at the functional level with the ability of an mGluR2 agonist to downregulate neuronal firing. And that ability is lost in the P rat. And there it is in red in the NP rat, which, you know, has mGluR2. And here's the behavior of the mouse. This is moving to another species. And for some reason the colors are wrong here, I apologize. The GRM2 mice drink more alcohol, and also the difference in alcohol preference in these knockout mice is not due to taste aversion as measured with saccharin or with quinine, or taste preference is measured with saccharin. And this is in an operant paradigm. This is data from Marcus Heilig's lab showing that that an mGluR2 antagonist, this is in Wistar rats, is increasing alcohol consumption. Here it is, the increased consumption anyway on day 2, day 3, and he's developing a lot more data. But it's not really a surprise to anybody that mGluR2 might alter alcohol consumption. So now I'm just summarizing. This is what we found. There's selection of a functional GRM2 stop codon from an ancestral population, which we now know has a allele frequency of about 8% for the stop codon. These are Wistar rats that everybody's using for neuroscience studies on glutamate, and about 1 in 6 of them actually carry a stop codon for mGluR2. And then the thing is selected to high frequency, actually 131 out of 131 preferring rats are homozygous for it, and to low frequency in the non-preferring rats. None of them have it. It's amusing, isn't it? So here are the conclusions of this talk: that addictions are ancient, moderately to highly heritable. They're context determined. There are alleles of all different types, including frequency and effect size and location in the genome, that are altering these complex behaviors and others. There are common functional variants that are modifying vulnerability in ways that are consistent with effects on mechanisms of addiction. So, you know, we're concerned at this course with emotion and impulse control and brain connectivity, not much here about metabolism, but variants that modify vulnerability to addiction have been found that work in each one of these different categories. Gene environment and gene bioendocrine interactions occur. The gene effects tend to be stronger, closer to gene action than further away from gene action. Rare alleles are discoverable by deep sequencing, and it's a way of getting at this problem of the rare and uncommon alleles in the in that genetic heterogeneity model. And genomics is opening new ways to identify addiction genes in old animal models. So there are a number of challenges that we're facing. The phenotypes that the clinicians want to stick us with are common in diagnoses with diverse causes, and the allele effects in that kind of phenotypic context are small and very context dependent. Meanwhile, the neuroscience-based approaches, which everybody here should be championing, are impeded by their cost and access, but also there are a lot of people who really don't understand what you're doing and what you're going to be doing, and you have to help them to understand why it is that brain imaging needs to be done, and why their clinical science needs to be connected better to the neuroscience, rather than you trying to adapt your neuroscience perfectly to a clinical science that is inherently defective in its conception. And so here are some collaborators. And Laura Bevilacqua is discoverer of the stop codon in HTR2B. Jifeng Zhao did the work on the GRM2 stop codon. Thank you. And we're now 12 minutes behind.
Not bad.
Not bad.
So have you found any rare variants for the metabotropic glutamate receptor in American Indians or in the Finnish population?
Not yet, but we haven't screened thousands of people. And when we screen thousands of people, we will find stop codons. We have found variants that are of some interest, but we don't know if they're functional. We have a haplotype of GRM2 which appears to be predictive of vulnerability. Jifeng has been studying it, but I've been discouraging him from even attempting to put this into print because we want to be at the level of functional locus really before we do that. And and he's been there before with his work on MPY. But no, I agree with you. It's just what needs to be done. And it could turn out that, and it looks like this way, it could turn out that severe GRN2 variants are rare in people. Maybe not, though, in some population, and that's another aspect of it. A lot of our work has been in American Indians and in the Finnish founder population. There are other special populations around the world that we would really like to sample in much greater depth. It's really impossible to to do it one gene by one gene, though. Ultimately, the way forward is that hundreds of labs do genome and exome sequencing, and by doing that, tens of thousands of individuals are collected. In order for it to work, though, those sequences have to be put into databases. And as soon as it's put into the databases, then a lot of the same discussion that we were having earlier about potential misuse of the data begin to arise again, because if your genome sequence or even a series of genotypes are in a database, and along with that sequence and those genotypes, there's any phenotype, you are matchable to that very simply. I mean, it's been shown as a recent paper on this that even if there's just a number of identifiers such as your place of birth and your age, your sex, and a few other things, that it's actually highly feasible to home in on a particular person. That's the identity of that person who is in that study. But if you have thousands of genotypes, or if you have any amount of sequence that's in a database, then the arrow can definitely be pointed right at you or to a member of your family. And the problem of being able to identify a near relative by virtue of your data in a database is something that people don't really appreciate, but it's happening all the time in forensics. So in forensics, most states, including my state, Maryland, now will take a DNA sample if you're accused of a crime. So you've been arrested for some crime. They collect your DNA sample. Your sample is then put up into a database. It can then be matched to a blood relative of yours very easily on the basis that 50% of the markers match. It doesn't have to be 100%. In fact, you can predict what the degree of relationship is quite well. And so that lets anybody know that in this second-hand fashion that you're potentially connected to something. And think about the way that it works. You're in a database, and I can tell you that if the sequences are in these databases, they're going to be used this way. let's say that CSI Miami goes out and they collect a sample from a crime scene, and that sample, 50% matches the pattern of your brother, which was entered into a DNA database. That points the finger at you and every first-degree relative of your brother. And it also points the finger to second and third-degree relatives, because that's how good the technology is today. You are implicating whole SIB ships and in constellations of related individuals through a single person being in a database.
So does GINA not offer protection? Is that just to the individual? Does that not extend to the family?
GINA could be remembered as Francis Collins' greatest accomplishment. This is the Genetics Information Non-Discrimination Act, and what it does is it introduced by Louise Slaughter and others, I can't remember, but it protects people from having genetic information used to discriminate in employment or insurance, at least in any overt, detectable way. So you can't in any overt way use the information that somebody has the Huntington's mutation, not to insure them or not to hire them. On the other hand, it has big holes in it, like all of these laws. It doesn't really deal with the problem that there are other assays for many of these genetic diseases other than the DNA. Yeah, like at the transcript level with the recent patenting the gene issue, and it does not deal with with this problem of criminal forensic identifications, and it just doesn't deal with the whole problem that some people do not want to know. And furthermore, once you're dead, Chris, what they probably get. My advice to you is to get yourself cremated, because what can happen is that once you're dead, you lose all rights. There are no rights at all that the dead have. The dead have no rights, and you can have your tissue specimen analyzed as this woman who thought that she was Anastasia, and who she'd had a biopsy, and so that was analyzed. It was proven that she was not a Romanov. Or they can get your actual tissue and sequence that, and they can find out all kinds of interesting stuff about you. And you have no rights.
And your relatives, too.
Because you're dead. Yeah, I think that the Henrietta Lacks thing is wonderful. I was just checking my email this morning, and there was an email from Francis Collins about that, announcing the decision. But I don't think that GINA, as wonderful as it is solving all of the problems of the fallout from this type of massive genomic information. Can I do one more before if there are any more questions? Thanks, everybody.